Hello everyone. So in this video we'll be doing about a biochemical characterization of transforming principle. We already know that this transforming principle, okay, it was given by the Frederick Griffith, okay, when he did some experiment with Streptococcus pneumoniae bacteria. So now the three scientists Oswald Avery, Colin McLeod, and Maclean McCarthy, these three scientists, in order to see whether it was DNA or RNA or protein, which biochemical is actually responsible for transforming the R strain bacteria into virulent form. What is that? Whether it is DNA, RNA, or protein, which biochemical? Okay, so what did they do is they performed some experiment. At first, um, they okay the wait heat killed S strain bacteria. The heat with this heat killed S strain bacteria, they subjected this heat killed S strain bacteria at first with proteases and RNAs. These two are nothing but proteases and RNAs. These are enzymes. Okay, these are enzymes that digest or kill protein and RNA respectively. That means proteases are the enzyme that digest protein and RNAs are the enzyme that digest RNA. Now this heat killed S strain bacteria in the cell of this bacteria, okay, which was heat killed, okay, this bacteria Streptococcus pneumoniae, the biochemical uh, that can be present inside the cell can be protein, RNA, and DNA. All the three biochemical can be there in the cell of this heat killed S strain bacteria. So at first they subjected this heat killed S strain bacteria with proteases and RNAs. So what happened if this S strain bacteria have DNA, RNA, and protein inside their cell? Now when they are subjected to proteases and RNAs, that means RNA and protein will be digested, only DNA will be left. Okay, only DNA will be left. Now after that, what did they do is, now the only DNA is left in the cell of S strain bacteria because this S strain bacterial cell was subjected to proteases and RNAs that killed RNA and protein, that digested RNA and protein. Now DNA is left. Now when only this DNA was left, only this biochemical DNA was left, this cell, this heat killed S strain bacteria plus R strain bacteria, which was non-virulent, okay, together they were injected into mice again. Now what happened? To their surprise, mice died. So now what they can conclude is, it is actually DNA only that is acting as a genetic material, because when RNA and protein was already digested, DNA was only left in this heat killed S strain bacteria. But even if only DNA was there in this bacteria, this DNA was transferred to the cell of this R strain bacteria and made that R strain virulent, transformed this R strain into virulent form. So the mice died. Okay, so that is how they concluded. Okay, these scientists, they could able to conclude that it was actually DNA which was acting as a genetic material, which was acting as this transforming principle. Okay, In order to prove that it was DNA which was transferred and made this R strain transformed into virulent form, they again performed the same thing, that again some of the heat killed bacteria, okay, bacterial cell which contain DNA, RNA and protein, because every cell contains this DNA, RNA and protein as a biochemical, okay, inside the cell. So now what did they do is they subjected this cell with the enzyme DNase. Now when proteases and RNAs are enzymes that digest protein and RNA, and now DNAs are enzyme, these are nothing but enzyme that digest DNA. When they subjected this heat killed S strain bacterial cell with the enzyme DNase, DNA was digested, only RNA and protein was left. And what did they do is, again, when this DNA was digested, okay, after that, along with R strain bacteria which was non-virulent, okay, they again injected to mice. Now mice was alive, okay. Sorry. Now see, this was also heat killed S strain. R strain which was non-virulent at first, this was also non-virulent. When this cell contained only DNA, okay, then the mixture of these two, when injected to mice died. That means it was DNA which was transferred from this cell into the R strain cell and made that R strain cell into virulent form, which was then able to synthesize polysaccharide coat and become virulent, okay. But here, okay, the S strain, heat killed S strain bacteria plus R strain bacteria, when together injected into mice was alive, because here the biochemical was RNA and protein, DNA was already digested. So that means it inhibit that transforming principle. When DNA was digested with DNase enzyme, that inhibit the transforming principle. So they can again conclude that it is not RNA and protein that is being transferred into the cell of R strain and transforming this R strain into virulent. It was actually DNA only that was transferred from this cell, okay, to the R strain bacterial cell and transformed that R strain bacterial cell into virulent form, okay. So this way it was the experimental proof, okay. It was done by Oswald Avery, Colin McLeod and Maclean McCarthy. So they showed that it was DNA which was genetic material which was acting as transforming principle, okay. All right, thank you.